clathrin vesicles. Again, this is something that wasn't really understood when I was in school. The magic of these things is that they can force the vesicles to form. I mean, it's not just like something comes and bumps into the membrane and it wraps around it and then takes it inside. That's not going on, okay? What is usually happening, in most cases, there's some sort of protein receptor in the membrane, okay? And when something that they like docks with them, they actually trigger a process underneath the membrane that starts this clathrin process going. And what clathrin is, it's a three-pronged protein that has sort of an arch shape to it. And these things start to crystallize together, and you can see if they're bent, when they crystallize, they're going to start to form a ball. Okay? And this is how this curvature is starting to form. Okay? It's these proteins on the inside are forming this structure, and it wants to form this ball and close it up, okay, as it crystallizes. And so you get a greater and greater curvature until you finally get a circle. And then there are several types of proteins that will actually snip this off and let the vesicle go. Okay? And then once it's inside, most of the time the clathrin will just sort of dissolve back off and leave it alone. Okay? And then this could be a food vesicle or something, and now the lysosomes fuse with it and digest things and things like that. Or a peroxisome will grab it. Okay? But that's the interesting and I like, these things don't have minds, so something chemically has to be doing something. It's all chemical, physical processes, so you have to wonder, what's the protein or what's the chemical that's causing these things to happen, causing these things to form? So you have to start thinking that way, okay? Because most of these things are just not banging around. So, but that's clathrin-coated vesicles is a very common way for cells to bring things into the cell. Okay? I think if you remember that, that's enough for me. Okay? This is the way it looks under the electron microscope. This is the way they look even bigger magnification. And you can't really see the separate proteins in here, but they form these mats like this, and then what they really want to do is fold up into a ball, okay, because of their bent structure. So this is what the underside of the cell membrane looks like in cells that are using clathrin vesicles to bring things inside. And then if you get a bunch of them, there's a sort of a limit to the size because of the size of the curvature of the protein itself. So you're not going to get huge vesicles or vacuoles, okay? But you're going to have these really tiny, tiny microscopic vesicles like this. Okay? And this, again, might be beating a dead horse, but so you have these different chains like this, okay? They will fold up together to form these hexagonal structures, but because it's bent, it has this angle, okay? It will give you a round ballish structure in the end, okay? When it finally crystallizes and hardens together. All right, now we get to the meat of this last part here. We've only got about 5 minutes, so we won't get too far. We'll finish up on Thursday, and then we'll have Tuesday to do a lot of review, I think. Okay?
Spring break?
Well, Tuesday after spring break.
So our test isn't...
Yeah, you want to meet us here? You want to meet us here?
So our test isn't the Tuesday as soon as we come back?
No, it's Thursday.
It's the Thursday.
Oh, okay, I thought it was a Tuesday.
We'll have one sort of empty day, as well as lab. Okay, we'll be able to do like a practical test in lab. So that'll be a lazy week for me. A review and then a test. All right. I think you'll find that when we talk about how DNA is made in the protein, I think you'll find this pretty interesting. This is why we're here, after all. And this is the amazing part about how the more you see how it works, the more you see what can go wrong, and just the more you should be happy to be alive and anywhere near normal. Okay.
Physically, at least.
Yeah. This was jokingly called the central dogma back in the 50s and 60s after DNA was discovered, because they thought that DNA was the secret, and it is for us nowadays. Okay? But remember, this is just 1 2 generations ago, nobody really knew what the genetic code was, where it was. They didn't know what a gene was. Okay? They didn't know why some genes caused a disease, and other times you had to have several genes to cause a certain condition. They weren't sure if all heredity was like marbles, where you had a green marble, a red marble, and a blue marble, and whatever you got from your parents, you had those, okay? And now you had green marbles and red marbles and maybe a purple marble and a yellow one from your dad, and you had different traits like that, and that would express themselves. Or whether some of the marbles would blend, and you add a yellow one to a blue one, you get a green one. Okay, they didn't really know how traits were transferred. Okay, and this about the time I was born, in 1954, is when DNA was discovered, and so and in a kind of a weird way too. There was a lady named, her last name was I think it was Frances Williamson. She actually did the X-ray structure of the DNA molecule to see that it was a double helix. Okay? And then the guys that got the credit for discovering it snuck in the lab and looked at the pictures, and they published. And they're the famous ones now. And she should have got the Nobel Prize, but she died before it was given. And so, but you all hear about Watson and Crick, right? Well, they were pretty bright guys. They knew what they were doing. They knew what to look for.
They're clever.
They couldn't have done it without her work. They've never given her enough credit. So DNA, in my lifetime, all of a sudden becomes the secret of life, and they find out the bases, because they really didn't know. They could isolate chromosomes, and they could analyze it, and they knew there was phosphate and ribose in there, and they knew the bases were there, but they didn't know how they were hooked up. Okay? They didn't know what the backbone was. They didn't know whether it was covalent bonding holding things together. Say an adenine and a ribose, and maybe the 2 phosphates were hooked together, and the chain was made that way. Okay? They didn't know. Okay? And that's why they needed that structure, the X-ray structure, to tell. Nowadays, with the instrumentation we have, you can figure that out a lot faster. But back then, they needed the X-ray to see that it was a double curve, a double helix. Okay. Anyway, we all know that DNA can make a copy of itself. It usually does that one time, okay, and we call that replication. Okay, and then it goes into prophase, and the cell splits, and blah blah blah. The rest of the time, when the cell is doing its job, it has to use that code to code for things, code for proteins. Okay, and to get to proteins, we first have to make a base copy of the DNA. And then that goes outside the cell, and that's read by the ribosome, which then puts the amino acids where they should be. Okay, and I'm going to show you how all that process works, probably mostly next time, because we only got about 2 minutes. Okay, but that's called transcription. When you make RNA, it's called transcription. When that RNA is then made into protein by the ribosome, that's called translation. Okay, and I think I am going to stop here. That's a good question. When the DNA is used to make RNA, that's called transcription. When RNA goes outside the nucleus and goes into the ribosome, and the ribosome then uses that template to make protein, that's called translation.
Okay.
Okay?